Hey all, so today we're going to be looking at D1.3, which is mutations and gene editing, and this is related to continuity and change. So how are things changing? How are they staying the same? This is in the Oxford textbook from page 593 to 605, and what we're trying to answer is how do gene mutations occur, and what are the consequences of those gene mutations? So firstly, what are gene mutations? So DNA has strands for connecting strategies for correcting errors in copying of DNA to prevent changes in the code. And if a change occurs, this is random and it's called a mutation. This can cause different amino acids to be coded for the protein, and that can potentially change the shape of the protein. So an example of this is sickle cell anemia. We'll see the specifics of that in a couple of slides. But what has happened here is a base mutation has occurred, and that has changed the outcome of that cell. So a substitution mutation is where, so this up here is the original, and a substitution mutation here is where a change has happened so that C has changed to a T, and it then may produce a different amino acid. An insertion is where something has been inserted into the space here, and that's going to shift things over. It's going to cause a frame shift, and that usually will cause problems. And then a deletion is where one of the bases has been removed and shifting things. That'll cause a frame shift as well, and that usually happens problematic to the production of the protein that we need to be making. And so in a deletion, we have two breaks happening in the sugar phosphate backbone, and in an insertion, we have one break happening. So the consequences of the mutations, some mutations can happen and they're silent. So they result in the same amino acid. A lot of amino acids have slightly different code, like there are three codons. Their codon segment has a bit of variation. That's optimal, potentially. If we have a missense, that means it can change the amino acid, which can critically change the protein. And many genetic diseases are resulting because of this problem. And then a nonsense is where the amino acid code is changed to a stop code, or it's not readable. It's changed to a stop code. So that prevents it from making the full protein because the ribosome can't read it properly. With sickle cell anemia, what's happened is there has been a base substitution that has occurred. And so instead of CTC, we have CAC, and that results in the RNA going from GAG to GUG. And that means glutamic acid is no longer being coded for, it becomes valine. And because of the structure of valine versus glutamic acid, the bonding, meaning like the hydrogen bonding, doesn't occur similarly. And so then instead of it forming a round shape, it flops open, and red blood cells then subsequently don't have the proper shape. So when we're looking at reading this code, a lot of small mutations or numbers of mutations can be beneficial, but they're much more likely to be problematic. So natural selection is going to select for beneficial substitutions. Large numbers of base substitutions have happened in the past in non-coding regions, and these are called single nucleotide polymorphisms. They're used in DNA testing because they are unique to you and your own family. So my SNPs will be slightly different than someone in the class's SNPs. But your SNPs, these single nucleotide polymorphisms called SNPs, can be related to diseases. So a lot of autoimmune diseases, you can see in people's SNPs that they will have autoimmune diseases, such as Crohn's disease, for example, or rheumatoid arthritis. So most major deletions or insertions will almost always stop protein function or cause protein function problems, whereas minor changes, like one or two bases, may cause proteins to have problems or to stop functioning. And then we heard this word on the last slide, frame shifts, where the codons shift over by one or two, will almost always cause amino acids to change. And if three bases are added, then a whole new codon has been added. So that's not a frame shift, but if a new amino acid is in there or deleted, then that can still cause serious problems. So mutations are caused by chemicals, radiation. Mutations are all the source of variation. They will create new alleles, but and random changes are usually neutral. Variation is necessary for natural selection, but it's not possible to mutate predictably because they occur completely randomly. So it's not related to whether or not the trait would be beneficial. Some sections of DNA are more likely to mutate than others because of their position on the chromosome. And if a germline cell is mutated, that's the gametes, it will pass on to its offspring. It is very problematic, usually, when germline cells mutate, because those mutations can be quite dramatic. This occurred in the Vietnam War, where Agent Orange was used as a chemical defoliant of plants, and it caused extreme mutation in the ovaries and testes cells of the humans exposed to this, including American military, but also the Vietnamese people, and caused extreme birth defects. So when somatic cells, those are your body cells, mutate, they're going to die with the individual. But that can result in cancer. And so if you are being exposed to X-rays or ultraviolet rays, for example, what can happen is your proto-oncogenes, which are cells in your body that prevent cancer from forming, are then mutated to become oncogenes, and then cancer will grow. So your body has proteins to prevent DNA problems. And so BRCA1 is a tumor suppressor gene, and this works to create a protein that repairs your DNA. But if this gene mutates, it cannot make the protein to fix DNA properly, and it will lead to breast cancer, ovarian cancer, and prostate cancers. There are 20,000 variants of this gene, and there's a BRCA exchange database, so you can pull up information about these different 20,000 variants. And some of them will increase cancer risk. So in some cases, rare cases, up to 80% over the lifetime for women and breast cancer. And so in some cases, if you test positive, and you'll know this, but you should be genetically tested, potentially, if that's something you want. If your family members have passed because of breast cancer, ovarian cancer, or prostate cancer, then you might be a carrier for one of these genes that would potentially cause breast cancer over a lifetime. And then breast tissue could be removed to prevent that long term. Some genes are highly conserved, so they don't mutate much over thousands of years. And the hypothesis for this is that these are coding regions needed for critical body functions, or non-coding regions that, while we don't know yet, these may have critical functions. So although certain areas mutate more than others, for example, the HACNS1 gene is mutated in humans, and not in birds and mammals. This has to do with our hand and our wrist development. And so that has not changed much for other organisms. So knockout mice, if we want to know what do genes do, genes have a start codon at the beginning, and then random codons throughout, and then a stop codon, and this is called an open reading frame. And gene knockout removes a gene segment, to see what the consequences are, so we can know what that gene does. So the gene is inactivated, and then the embryo is cultured and grown in a lab mouse surrogate. That offspring has one allele that doesn't work, and then 2 knockout mice with one functioning allele are crossbred, which results in offspring that have no functioning genes for that. And this is called a knockout mouse. And then we can see in these mice babies what's going on without that gene. So CRISPR-Cas9, CRISPR is a region within prokaryotic genomes that is used to detect viral sequences and targets them for destruction with the Cas9 enzyme. So viruses like to inject their genetic material into prokaryotic DNA. And so CRISPR-Cas9 is a way of controlling that for prokaryotes. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. You don't need to memorize that. And Cas9 can find foreign DNA in the cells using some guide RNA, which contains a spacer, and the CRISPR array connects to the virus and cuts out the foreign gene. So scientists can create their own guide RNA with the sequence of DNA that they want to identify, connect it to Cas9 so that segment can be cut out once identified, and they use a modified version of Cas9 so only one cut is made, and Cas9 is also able to attach a new segment of DNA into the cut region. So to summarize, mutations are random and they lead to genetic variation. Typical mutations are neutral, but can lead to problems in protein creation. There are insertion mutations, substitution mutations, and deletion mutations, and insertion and deletion are going to create frame shifts. Missense, nonsense, and same sense changes. Same sense then is going to result in the same protein being made. Nonsense is going to result in no protein being made. And missense is going to be a different amino acid is made, so that protein might look different. Frame shifts can lead to change in amino acid coding. Knockout mice allow us to know what genes do, and then CRISPR-Cas9 allows genes to be modified in our DNA. And that is all for this video. Thank you very much for watching.